All right, so I will be talking a little bit about decoding today. Before I proceed, these are the two recommended reading that I would suggest. I'll put the papers in the Slack channel. These are really two good review papers for kind of like reviewing how people think about going from neurons to behavior. And the second one is from Jim, and this goes into details of how visual object recognition might be getting solved in the brain. So just kind of to revise what Puya has been talking about, how I kind of think about systems neuroscience in general is that you have an input to this, where is this? Yeah, you have an input to the brain, and then the brain represents the input and then does some behavior, like we make some motor movements. So for example, you can think of like you're thirsty and you want to drink water, and you see a glass of water, then you go and pick the glass of water. But let's say you see this image, then you will not pick that glass of water because it's empty. So basically, depending on how these kind of visual images are represented in the brain then goes on to read that and produce a particular behavior. So if you're in systems neuroscience, you are either going to study the encoding models, like how the visual world is represented in the brain, not the visual, how the world is represented in the brain, or you're going to study the decoding model, how the representations are then used for behavior, or you're going to study the combination of the two. And if you're not doing that, if you're going from the stimulus directly to the behavior, you're probably in the psychology department. Okay. So one important thing is that for this entire setup, one of the most important thing is behavior. You have to quantify this behavior very well to sort of have a good sort of gauge on how good your decoding models actually are. So this needs to be defined very quantitatively to evaluate how good the models are. And I will basically like emphasize that good here means good models of the brain. I'm not talking about high performing computer vision models. I'm talking about models that actually do or try to mimic the brain. Okay. So in terms of behavior, as Puya already mentioned, that typically people have been thinking in terms of two pathways, the ventral stream and the dorsal stream. So there is a lot of studies that have gone into dorsal stream, and I will actually touch base on them during the psychophysics tutorial. It kind of lends itself to these kind of psychophysics tutorials, but today, especially because I'm now working with Jim and we all always record from IT and V4, and so I'm going to talk a little bit about ventral stream because I kind of know the history a little bit better maybe. So I'll talk about and also this is approved by Tommy because he has this code in his paper that understanding how biological visual systems recognize objects is one of the ultimate goals in computational neuroscience. So I'm going to talk about the object recognition problem. So let me kind of motivate the problem because this is a very natural thing that happens every day. So here I am standing in front of the Harvard T-stop, and this is a scene that's being bombarded on my retina. And I don't see the whole thing at once. I will basically parse this scene based on some eye movement. So I'm probably going to look at this and say, oh, it's Harvard Square. Oh, there's a newsstand. There's a guy walking. There's the bicycles. There's like a car approaching. So that's how I'm going to parse this scene. And so this is a very natural movement. I agree that there are microsaccades that are basically jittering a little bit of this stimuli. But more or less for 100 milliseconds, these are stationary images that are falling onto your retina. And I don't need to give you the context. I can just ask you to fixate and repeat these images one after the other for 100 milliseconds, and you can still tell me what objects are there. So that's kind of the motivation why we study this paradigm, because this is exactly what you're seeing, this is exactly what is falling onto your retina. So we have done some research, a lot of people have done research on this field, and we kind of sort of know the areas that are implicated in this. This is the ventral visual stream that we were referring to. So in terms of, so I was thinking like how should I kind of like organize this tutorial. So what I'm going to do is I'm going to give you an example of how our lab studied this phenomena and how our lab built a decoding model of this behavior. So I'm just going to take you through the steps. So that might make it easy to understand how one could sort of approach the problem. So the most important step is you have to define and operationalize the behavior. If your behavioral metrics are not good, then the whole project kind of falls apart. So in this case, we decided to go with a binary discrimination, object discrimination task. It's a pretty simple task. An image will come up in the center, and then there will be two objects, and you have to choose which object was presented in the image. So for example, like this. So there was a person in the picture, then something like this. It's a bird. I hope you guys got it. So this is like for 100 milliseconds. So this is pretty much the task. There is a monkey or human or machine. They just look at this image, and then this 2 option comes up, and they have to choose. And so in this way, we can run like thousands and thousands of images. This is a very standard task, so it's nothing new about it, really. Now, the second question is, what is the behavioral metric? So I'm going to show you one behavioral metric here. This is a confusion matrix. For those of you who don't know that, I'm just plotting the performance of the subject, whether it is human or monkeys or models, for each object when they were tested against all other objects. So this will be like camel versus dog. That will be like camel versus rhino and stuff like that. So these are 2 different species, and they look very similar. So that's kind of the second question. So if you are part of an EEG lab or fMRI lab, you are probably going to use a human as a subject. We wanted to build decoding models not from EEG signals, but from neural signals. So we need to choose a particular model animal that we can run. So the criteria is that the model animal should be consistent with the ultimate animal that you're trying to study, which is human in this case. So I'm not going to tell you, but one of them is a human and one of them is a monkey, or one of them is, this one could be monkey, this one could be human. But they look very similar, and they have very similar confusion matrices, confusion patterns as well. So in summary, like we have published this study at multiple grains, comparing different levels of object recognition behavior between monkeys and humans. So we choose monkey for that. That's why we didn't go with the mice. And I think that's important to consider during sort of choosing behaviors, which animal models you can tackle. The next thing is that once we have chosen monkey and a behavioral metric, we can now decide like where to find the decoder. And it's like this step cannot be completed without the encoding part, and that's why I think Puya went first, because people had to probe different parts and understand what an area is basically encoding. So where to look in the monkey brain? So there has been decades of neuroscience trying to probe this thing, and it's not only correlative evidence of like collecting neural data, it's also lesion studies, so causal perturbations. You perturb IT, you have deficits in visual recognition. So you kind of know that these areas are kind of critical, okay? So based on research for decades, we kind of have a model of like how the ventral stream might be working. And it goes like this. So the image comes in to the retina, and then it gets transformed. So these, you can think of these as patterns or responses of these cells here, and it just gets transformed from one step to the other. So from LGN to V1, V2, V4, and back to IT. And the other interesting thing is that when you have different images, the pattern of activity in IT changes. So it does for every step. But I would like you to now concentrate only on IT. So what happens is that you keep on changing the images, and the pattern of representation keeps changing. Okay. So what I think it was Chao Hang and Gabriel also was part of this research. They found out that you can actually decode the categories of objects based on activity in IT. I think it was like 2005. So based on this study, now we know, okay, if I want to search for a decoder or search for area that a decoding area might sort of read from, here is my first guess. I'm going to guess it's in IT. So IT has different size, and they all fire like different spikes, different spiking patterns based on different objects. And typically what happens is that there is like this firing around 70 to 170 that Puya was also mentioning, where most information about the object kind of lies, okay? And this is a result figure from their paper showing that you can actually, with around 256 sites, do a very good job at categorization. Of course, this curve will depend on the kind of images that you have used, the kind of tasks that you have probed the system with. So don't take this graph very seriously. Take the sort of like the qualitative evidence from this that you can categorize, okay? So IT is fixed. Now the problem is that previously we did single electrode recordings. I'd been critical of it yesterday. Probably shouldn't have been so much critical of it, but I still think these new techniques of using Utah arrays, where we don't consider single units but like many units at the same time, this scales up the process by a lot. And I think this really helps, because at least the results show that the code is in the population, it's not in single neurons. You can do this in the single neuron in the sense that you can keep recording single units over and over again, and then you can make a big pool of neurons. That will also work. But that needs 3 years, and this will take 3 months. this is how our data collection rate has improved from 2005, which was a single unit recording study, till now we are using arrays, and it's a huge exponential improvement. Okay, so how do you approach this problem computationally? You can think of each image producing a vector of responses across IT. So these are kind of the features of IT's model of the world. So anytime an image comes in, you get a number of firing rates from each neuron. So it could be 100 to 1,000 neuron. And if you record a lot of images, you get a huge matrix like this. Okay, so now you have shown different images. So the x-axis is like individual images. And for each of these images, you get a huge matrix like this. So the computational problem now is that you have a behavioral metric or a behavioral sort of measurement that looks like this. And you have to find a model that goes from this, links this big chunk of matrix to this, right? So this is the decoding problem that you're trying to address. So you needed the encoding study, you needed the proper behavior, and now you're at a level where you can start asking, can I build some decoders that tries to be consistent with the human behavior or the monkey behavior? And the specific parameters of the decoders are important. I want to emphasize that this is not a qualitative study. This is not a study that will say IT works somewhat to do object recognition. We are trying to make a claim that you need 60,000 neurons, X number of dimensions to predict 70% of the explained variance. This is the kind of grain at which I think we are trying to push the field, okay? That's why the exact parameters are also very important. So here's a graph from one of the papers, a figure from one of the papers, and they have compared different decoding schemes trying to predict human behavior. So the consistency measure is also sort of something important, and that I actually did not know before joining this lab, because I typically used correlation as the measure of how similar two things are, but there is a bound to that correlation, and the bound is how, for example, if I'm a model of another human, I need to know how consistent that human is to himself or herself. So that kind of sets the limit of my performance. So this human-to-human consistency is a very crucial, important term. So anytime you're building a decoding model, that is something that you really need to pay attention to, okay? So here, the results are that if you take models that are pixel representations or V1-like or older computer vision hypotheses, or for example, V4-based models that are basically responses of V4 neurons, they don't do as well as IT-based hypotheses. And even for IT, there is a specific set of like algorithms that typically work very well. And you can see that from correlational decoders, I don't have enough time to go into the details of it. I will try to elaborate a little bit in terms of what SVM kind of geometrically sort of like does. The other important thing is the time at which the features were computed. So this was a static model in the sense that we just averaged the responses from 70 to 170 to produce any of those vectors for each of these images, okay? Now, I just want to give you like a feel for what we mean or what SVM might be doing in this case. And the idea is that whenever I have different images of the same person, for example, like let's say this guy Joe, and these are axes of different neurons in IT or any visual area, for example, every image will have one particular spot in this multidimensional space. So here's one image of Joe, and then there are many images of Joe I can show from different angles, from different sizes. You know, and it kind of like, it has a space that it covers within this multidimensional space. And you can call it Joe's identity manifold for this particular, whatever area you're recording from, okay? And it turns out that if you look at V1, this is kind of what it looks like. So if you have Joe versus, I don't know, Harry, individual 1 versus individual 2, the manifolds are kind of tangled with each other. You cannot really separate two different individuals or sometimes two different objects if you're just looking at V1 representation. And the idea that was proposed by, I think, Jim and Dave Cox at that time is that, or it might have been reformulated from previous ideas, that object manifolds might be getting untangled as you go up the hierarchy. So you can think of this hierarchy, V1 has a much tangled representation, and the tanglement like kind of decreases, gets untangled as you go to IT. So for example, in IT, you might have the manifold express itself kind of like a smooth manifold. And now, if you look at Joe versus individual 1 versus individual 2, it might look like this. And what the decoding model is trying to do, SVM is trying to do, is trying to put a hyperplane through it so that it can basically classify any image into either Joe or not Joe. Okay? So this is a very simple linear classifier, and that's the reason why it can be, it's like biologically plausible that a downstream area from IT could be just reading IT to go to behavior. So that's why we think that in terms of decoding, it's just a linear step away from the behavior, IT. Okay, so again, coming back to like the specifics of the model, I cannot emphasize this enough that these numbers are important. The number of dimensions that IT spans, like how similar is one monkey to the other, or how similar is one IT to the other, how you even go about even answering this question. I think that needs you to be more quantitative, to think about sort of like how many dimensions these kind of areas span and stuff. So that's why these numbers are important. So whenever I want to compare this to my own study, I want to know how many neurons I need to record to get to human level performance. So these numbers are important. Sorry, I think I kind of skipped the slide here. This is just showing that if you're going from 16 sites, you have some amount of correlation for all 64 tasks, and the correlation is higher for IT compared to V4, but it's not to the level of humans. As you increase the number of sites, you get to a point where you're equal, okay? So of course, this depends on the grain, the grain of the behavior that you have tested. So this behavior is somewhat sort of coarse, because I'm averaging all the images of a given object to get the behavioral metric. But the thing is that because the approach to this problem was sort of like leading up to this sort of conclusion, we can now ask, okay, let's make the behavioral sort of metric a little bit more fine grained. So let's go from object level to image level. So this is like the fifth sort of test of your decoder. So make the task harder and see whether the decoder still holds true. So philosophically, once you have a model, this is the job of the experimenter. The job of the experimenter is to design experiments that falsify the models. And unless you have a model, there is no falsification. Science just stays in the same place, and we all have our subjective opinions about things. So here is a case where you can do an experiment and say, look, Jim, like this model is wrong. And I think that's what I did, because I started doing this in terms of like image level performances. So now basically what I'm doing is I'm kind of splitting this metric up. You need a lot more data to get to reliable measurements like this, but you can do it. It's like Amazon Mechanical Turk or Monkey Turk. It's pretty easy right now. So instead of having like only average of all camel images, I have individual images. So here I've shown 10, but there were like hundreds of these. And you can have a huge matrix. And now you have to predict the entire thing, okay? So I'll give you an example of why I think that 70 to 170 millisecond decoder is wrong. So remember, the previous decoder said take the activity, integrate it from 70 to 170, just use that to predict what the object was. So I have recorded, right now I've recorded more, but this slide was made a little bit earlier, so it's 333 neurons, but still does the job. So let's say I show this face image. And what I'm doing is I have computed the average performance of the monkey for this particular image. So the monkey's D prime is like really high for this image. These are the same vectors that I was showing, but now they are computed per time bin, so per 10 millisecond time bins. So for every 10 millisecond, I have a vector of 383 elements, and then I use a decoder on top of that. The decoder is still the same. It's the SVM, linear SVM classifier, and I try to predict that given that activity, what is going to be the neural decoding performance, or what is going to be the predicted performance of the monkey. So what I see here is that the monkey performance is like really low, the neural decoding performance, and then at some point around 100 or so, it's like high, and it hits the level of the monkey's real accuracy that was recorded during the time I was measuring these. Sorry, go ahead. No. So the reason why, so we wanted to go lower and lower for other purposes, like we wanted to make it really kind of like tiny, the time bin, but the tinier you go, the noise increases. So we kind of did an analysis to understand, like, given an expected sort of effect size, what is the lowest time bin that we can go to with reasonable number of repetitions of images. So that's why it's 10. And so we need around 70 to 80 repetitions of every image to stay within a very low number. So that was a phase, and then I show this zebra, and then I can do this again. I get the recordings, and you can see that they're kind of like getting solved around here. Now you use another image. This is an image of a car. These images are picked that the monkey has the exact same decoding accuracy. But now you see that the response is going up and it's hitting it around maybe like 150, 180. So if I was just integrating from 70 to 170, the answer for this would not have been the same as if I was integrating from 70 to 170 for these guys. But that's sort of like a, sorry, go ahead.
When you say monkey accuracy, you were talking about the accuracy of the decoder?
No, this line, is that what you're asking? This is, so when the monkey was doing the task, this is the accuracy that it had to tell that this is a dog versus any other.
Can you stop showing the image after you make the trial for each such duration?
No. So this was done, so the images were shown for 100 milliseconds, and then the task was performed. So what happens is that the monkey sitting, images get shown for 100 milliseconds, then the monkey does the task, and the firing rates are recorded throughout. So I'm basically trying to see at what time in that zone the firing rate is good enough to give a behaviorally relevant prediction.
So you impose the time when the monkey has to answer?
No, so the monkey doesn't know about this analysis. So the monkey just knows that he's going to see this thing for 100 milliseconds and has to tell whether it's a dog or a house or dog or an elephant.
So, I think I just don't understand the x-axis.
The x-axis is time from the image onset for the neural data. So I'm recording the neural data, which is a continuous measurement. So I can record the neural data when I just showed the image, when the image went off. Sorry. Okay, go ahead. Or yeah?
Oh, yeah. Are you decoding using strong average data?
Yeah.
So what if we use single strong data?
So the problem is the single trial data has a lot more noise in it. So that's why we use trial average data. The interesting question is that when we do use trial average data, does the decoder suffer somehow? Like, I'm almost averaging out a lot of interesting phenomena that people talk about, like noise correlations and stuff. So because I'm averaging them out, is my decoder suffering? And so far it isn't. So maybe trial by trial data is not that important to build this kind of models.
I would presume that trial average data would be better for the
Trial averaged?
Yeah.
Yeah, it is because of high signal to noise.
But what about, so if you want to go a little bit more study, if you want to relate this to the monkey's behavior,
Yeah.
single trial data matter?
No, because it doesn't matter how you get the decoder, because you can get the decoder from trial average data. You can still use that decoder to predict the monkey's response for every trial, right?
Is this only on correct trials?
No, this is for all trials.
All trials?
Yeah, and you can even do this for monkeys that are passively fixating. It doesn't matter. So you can collect the data separately, and when the monkey is in the rig while we are recording, he can just be passively fixating. You can still get a pretty similar decoder. So it's a very automatic process that's in the head. So, yeah.
So then what about if they misclassify the dog as something that looks like a dog?
Yeah, so
Does this decoder look worse?
no, so the decoder would, so for example, this is not at 5, which is our cutoff. So at 5, everything, he got everything right. So for example, now this is at 3 to 4, so there are some incorrect trials in here. And that also shows up in the decoding accuracy, right? So the decoder is capturing the fact that probably the monkey is not going to always get the right answer. So you can do this either on a trial averaged way where even the predictions are trial averaged, or you can go every trial, use the decoder and say, for this trial, what's the monkey going to do? And I have some backup slides that addresses that, but I can show you a little bit later. Anyways, I think I'm kind of at the end of this, that this was hinting to the fact that, look, the decoder is wrong. And for example, we collected 5,000 images like this, and the time at which these images are solved are like really variable. It's like from 100 to like around 200. So it's like hugely variable. So recently what we did is that, so the question is now like, okay, seems like the old decoder was wrong, what's the new decoder? And now we have a new decoder, which is, I'm not going to go into details of it, it's just a leaky integrator. So the scheme has changed now, so we don't discard any time bin, we just integrate until the choice screen is shown. And it seems like that's a better decoder to sort of deal with this problem. And the important thing is that what is the time constant of this leaky integrator? So it's like around like 40 milliseconds. Whether that has some real value is to be seen. So just to make a point, like, so Puya talked about the encoding model, and here is a realistic model of like what the monkey might be using from IT to go to these behaviors. So if you remember, so I had a project in my mind where you can now know what kind of images the monkey will think where, or what kind of images of, let's say, a dog that you can show the monkey, where the monkey is going to say, oh, well, it's an elephant or something. I think that's a cool experiment because that clearly shows that you have information about the monkey that otherwise was not possible. So because you have the encoding model and the decoding model, you can even think of things like you show like a thing getting morphed, and you can predict exactly when the percept will change of the monkey, from a dog to human or dog to elephant or whatever it is. I think this is the end of my presentation, and you can ask me questions for 19 more days. Just to kind of like answer your question, I think this is the slide. So forgive me for this particular. So here's an example where I want to test trial by trial. Okay, so here's an image that has a, if you can, I mean, this is a little bit tricky to maybe see. There is a plane and a bird in this image. In this image, there's also a plane and a bird. You can't see it, but there, believe me, you have to believe me here. So there is a plane and there is a bird, and the choices are also plane and a bird. And the monkey on average is at chance because the monkey sees both of the objects and he's pretty confused. But because you can record, you can for every trial have a prediction of what the monkey might choose. This is typically known as choice probability, where you show an ambiguous stimulus and you can see how much you can predict the monkey's choice on a noisy stimulus like this. And you see that the chance level is at 50%, and with this kind of decoder, which is a leaky integrator to the choice screen, we are at 0.65. And this value, if you have been following the choice probability literature, is pretty high. This is like, but it needs a lot of neurons too, so maybe that's where we are getting the little bit of edge. But here is a comparison of laws of RAD IT, which was taking the chunk from 70 to 170 and was not looking until the end. So that's almost at chance. So you can do experiments to sort of falsify these kind of older models further, but there is an interesting space of images and experiments that we can still do to make this even like a more stronger test of this kind of model. So I think, I don't know if I answered some trial by trial questions that you had. I think that's it. Okay, I'll take some questions, but I think at some point, both Puya and I will take questions. So this is it.